criticized Graham in an ASM meeting because they called them massively parallel undergraduates. No, they're each on their own phage, and this is just the start where they're going to end up. Right? And so Lenny was a classmate of my kids at Montana City, where he started phage hunting, also at Helena High with many of your colleagues who are in the BRIC program. He's currently at Montana State University, double majoring in biology and music. And I've heard him play the cello, and I can just tell you that he's amazing. And he has so much to offer both those worlds, and I hope he continues in both.
Thank you. So, hello. So you've got an introduction to me, so I'll just start out. My timeline with phages, I started in 2016 in middle school with the phages program. Took environmental samples, you know the process. Sadly, I found no phages myself. Is the microphone working? Okay. Yeah, sadly, I found no phages myself, but I had a great time. I was introduced to a lot of scientific instruments, and I gained a lot of the basic knowledge of how these cellular processes work. I think that was really valuable going forward. This is me in middle school. And I had a similar experience in high school. Again, sadly, no, I didn't discover any phages, but it was a really good experience again. But I think the biggest impact that the Phages SEPA program has had on me has been the funding for a pipeline student project at MSU. This is me in the lab of Dr. Blake Wiedenheft, which is the lab that I've been working in at MSU for a while. This summer project that I worked on, I'll detail it a little bit in the next few slides, but at MSU I've also been really involved in music, as you heard. And yeah, it's been great there. So starting off with the project that I did that was funded by SEPA, the pipeline project. My mentor at the time was Tanner Wiegand, who was a postdoc in the Wiedenheft lab. So you're all familiar with the process of phage infection. Phage comes in contact with bacteria, introduces its DNA, which transcribes, translates all the proteins and components, and then replicates its own genetic material, DNA or RNA, I should say, which is then assembled into the virion, which breaks out of the bacterium. The bacterium dies, virus replicates. Bacteria, instead of going some of the routes with like phage ecology that we've been talking about, I went in a different direction with this relationship between phages and bacteria. And I studied some of the immune systems that bacteria use to defend against phages. We're familiar, a lot of immune systems in bacteria have been characterized for this purpose, but as far as I'm aware, we only know of one that's adaptive, and that's CRISPR. For those of you who aren't aware of exactly how it works, when the phage introduces its genetic material into the bacterium, a small section of that is taken and inserted, copied into the CRISPR array. CRISPR, just the acronym describes the architecture of the array. It's a cluster of regularly interspaced short palindromic repeats. So you get little bits, sequences from past invading viruses, different ones, that the bacterium can use to defend against those same ones in the future in a sequence-specific manner. It's a molecular memory. It's pretty cool. Cas just is CRISPR associated, so it refers to all the proteins associated with that system. This stage right here is called adaptation. The second stage of CRISPR immunity is CRISPR RNA biogenesis. All the proteins are transcribed and translated, and the CRISPR array is transcribed into CRISPR RNAs that are then assembled into interference complexes like this with all the proteins that go around the cell. They search for complementary sequences to their guide RNAs, which were originally taken from invading viruses in the past. When they find a complementary sequence, they chop up that DNA, so the phage isn't able to replicate inside the bacterium anymore. It's been shown in some cases that bacteria also, the question that we're trying to answer is, we know how a lot of this process works, but there's still some uncertainty as to how bacteria initially identify genetic material as being from phages so that it doesn't put its own DNA or RNA into its CRISPR arrays and do autoimmunity. That's the question that I'm trying to answer with this project. So we know in some cases in bacteria, they produce reactive oxygen species when they're infected with phages, which produces markers of oxidative damage on the different virus particles, and it results in fewer of them being infectious. We know that it produces distinct markers on of oxidative damage on the DNA and RNA of the viruses. So we were wondering, are those markers something that can allow the bacterium to distinguish between phage and bacterial genetic material so that it doesn't do autoimmunity, but so that its immune system is still able to function well in the future? So that's just what I'm illustrating here. So I guess that's our question here. The first thing that we looked at to answer this question is how can we simulate DNA that has been damaged through oxidation in the lab? We researched what DNA bases look like when they are subjected to oxidative damage. There's a lot of different things that can happen. Single-stranded breaks, double-stranded breaks, lesions. So we took PUC19, which is a plasmid that's used commonly in different scientific protocols, and we took a restriction enzyme that we know cuts it in 3 distinct places. Our procedure was treating this, before treatment with a restriction enzyme, we treated our plasmid with, which is a purified plasmid, not in a bacterial cell. We treated it with hydrogen peroxide of various concentrations and then subjected it to a restriction enzyme digest. We found that we were able to effectively, or I guess this is what we would expect just with the restriction enzyme digest. We get 3 distinct products. We found that we got disappearing products as our concentration of hydrogen peroxide increased. That was our evidence that we had successfully damaged the DNA in ways that we could quantify. Our next step to the project was transforming that DNA into the bacterial cell with a CRISPR array that's on a different plasmid. Because we want to see if we transform the DNA that is from the restriction enzyme digest without oxidative damage, do we see differential adaptation compared to transforming DNA in that had been subjected to oxidative damage? And sadly, or I guess this is what we would look like. We're able to design primers for our CRISPR arrays on our first spacer that we know exists and the leader sequence, which is always next to the CRISPR array. If new spacers are inserted, so if there's adaptation, that's going to get longer. And so we're going to be able to see that on an agarose gel. Sadly, we found that we did not see dramatic alterations in the amount of adaptation and in these CRISPR arrays. When we're comparing our controls, which is no hydrogen peroxide, to different concentrations of hydrogen peroxide treated plasmid DNA. Even though we didn't get the results that we thought we were going to get, and we received no evidence for our hypothesis, this was still a great experience. It introduced me to research at MSU, and it put me on an upward trajectory for different research experiences at MSU that I wouldn't have been able to access if I didn't have the funding from the Pipeline Students Program. So this had an incredible impact on my academic journey. Can't overstate how important it was. So after this, that was in the summer of 2022. After that, I received funding from the Montana INBRE organization. INBRE organized the internship in 2022, but the Phages funded it. The following summer, I was able to continue working with the INBRE program, and I got funding to conduct some research at Rocky Mountain Labs in Hamilton, which was an amazing experience. I learned a different set of skills. I worked mainly in an immunology lab. And I'm not going to go into as much detail on the work that I did there, but I'll briefly explain one of the experiments that I did there that was also, again, a great amount of skills that have been helpful since then. That concerns hepatocyte nuclear factor 4 alpha, which is a transcription factor that's important for the development of hepatocytes. We also thought, based on some screens that the lab had done in the past, I should say I was working with Sonia Best's lab at Rocky Mountain Labs, and I was under the mentorship of Adam Page, both incredible researchers. Previous screens that the lab had done suggested that this transcription factor also impacted innate immunity in different cells in the human body. And so previous research suggested that this might be due to different post-translational modifications, like places on the protein were phosphorylated or not, and that impacted how it differentially was relevant to innate immunity. We devised a scheme of site-directed mutagenesis where we changed individual amino acids in the protein either to alanine, which is like simulates the phosphorylation site not being phosphorylated in any case, or aspartic acid, which simulated the phosphorylation site being active in all cases. And we were trying to figure out if these different modifications to the protein impacted the way that it bound to the promoter for NF-kappa B, which is a much more well-known innate immune promoter. We found that using a luciferase assay, which this is here, the height of the bars basically just indicates how much activity at a certain promoter site there was, and the promoter site was the promoter for NF-kappa B. We found that one of the mutants that we created did have a significant impact on the activity at that promoter site. So that basically opened the door for a lot of future research into the specific pathways that happened there, and to see if replication of different viruses was altered in human cell cultures. I wasn't there for this step, but this was still an immensely important experience for my development as a researcher. That wasn't the only experiment that I did there, but it wouldn't make sense given the time to go through everything. After that, I received funding at MSU to continue research at the Wiedenheft lab, both during the school year and this summer, so it's going to be starting in about a month, which I'm really excited for. And I won't go through the experiments that I've been doing for my ongoing research there, but I do want to touch on the broader project because I think it's really exciting. So you're probably all familiar with we can test for COVID-19 in different ways. We have PCR tests and antigen tests. PCR tests test for genetic material. They're very accurate, but they're also time-consuming and costly. They require equipment that's not available in every scenario. Antigen tests, on the other hand, are very quick. Those are the at-home tests that you've done if you've ever done like an eyedropper or something on a lateral flow strip. They're quick, and they target proteins instead of the genetic material of the virus. But they also, because they target the proteins and not the genetic material, they are less robust to some cases of viral evolution. And so we're brainstorming, are there ways that we can develop a test that pairs the way that PCR tests rely on genetic material with the quickness and availability of rapid antigen tests? So one of the system that we looked towards was another CRISPR system. You're familiar with how CRISPR looks. This is our interference complex, which has our target RNA, and it has the guide RNA, looks around for target RNA or DNA. When it finds it in the cell, it normally chops it up, and that's how it does immunity against phages. We're able to take this out of the cellular environment and repurpose it for a diagnostic purpose, because we can program in basically whatever sequence we want as a guide RNA. And so for instance, we can make it look for a conserved region of a coronavirus RNA. And when it binds, this CRISPR complex is unique in that it has a part of the protein is allosterically activated when it binds to produce signaling molecules from ATP. In the cell, this is just a signal of infection that activates other pathways. But we were able to repurpose these signaling molecules by detecting them with ancillary nucleases that, when they bind to a signaling molecule, they perform another reaction that produces a fluorescent signal. I didn't take part in the development of the initial iteration of this test, but I've been a part of seeing how we can make it more sensitive, because although we know that it works, it's not sensitive enough for clinical applications yet. It requires concentrations of RNA that are higher than we would find in patient samples. So my role in the lab has been using bioinformatic methods to identify diverse effector complexes that might have different protein kinetics, might produce these signaling molecules more efficiently so that we can get a higher sensitivity or less background. Most recently, what I've been working on is seeing whether or not we can actually detect the depletion of ATP instead of the creation of these signaling molecules. And one of the things that got me into that idea was, if you'll remember from my summer at RML, I used luciferase assays a lot to assess promoter activity. Luciferase assays, when ATP is the limiting reagent, they produce luminescence, in a linear relationship with the available ATP. So as this complex depletes the ATP, our thought is that we can detect that depletion with luminescence, or with a luciferase assay modified. So we've proven that concept works, but right now we're we're trying to make it work now with relevant concentrations of RNA for patient samples. And so my role has been mathematically modeling this system so that we can get closer to the point where we're going to optimize this reaction biochemically. That's where I'm at right now. Hopefully this is what I'll be doing this summer, so I'm psyched about that. And that's it. A big thank you to all the funding that I've received, especially SEPA, Montana INBRE, USP scholars at MSU. And I've also had an amazing set of mentors and other people supporting me throughout this research journey. So thank you, and I'd love any questions that you have.
So out of your experiences, is there one thing that you think is similar or very different at the different stages? And do you have any advice for somebody who's just maybe, what would you do the same or differently?
A lot of my research experiences at the university level were similar. But I think one thing that could have made it easier and made me a more effective researcher sooner is being able to just consume a lot of literature effectively and fast so that I can get familiar with new projects and things like that, generate hypotheses that are my own, basically. So I think that's the one thing that I'd focus more on, is just getting better at consuming that media and generating my own hypotheses from it.
Like the project sounds cool. I look forward to my RNA CRISPR COVID testing.